Thank you. So thank you everyone for inviting me to come and talk to you today. It's really exciting to talk to other people that are linked via the NEQAS EQA scheme. We've been working really closely with them for the last 18 months really on EQA processes for our COVID testing services. So it's really nice to be invited to join one of their webinars. I'm going to talk a little bit today about quality issues around validating new COVID assays, a little bit about EQAs, the different technologies that we use, and a little bit about variants of concern. So in my current role, I assess assays that are submitted for validation verification, and I'm also chair of a group that looks at VOC assurance, so variants of concern assurance for coronavirus. And I've been working on the coronavirus response since January last year. So I've been fully involved in all areas, and I first began working for Public Health England with operational services for the regional labs there, and I'm currently working as Jenny mentioned for the Chief Scientific Officer in a national role with Test and Trace. So I shall begin the journey. Or not to the slide. There we go. So usually when we introduce a new pathology method, whether that's a single test or a whole service, it will take a long, a really good period of time. And it could be 6 to 24 months. And with coronavirus, it was a new service, a new test, and for some laboratories, a completely new experience for them to offer molecular testing as a service. So in a normal setup, you would identify the need for your improvement and investment. You'd look at the commercial options, you prepare business cases and look at your finance and clinical evidence to support your business case. Then you might go out to tender and then pilot the different options before you award to a supplier, and then put together your project plan and implement, and then once you're established, have a benefits review. What's happened in coronavirus is that in the last 18 months, we have month on month had to move goalposts, introduce new tests, change strategies in the UK. And I know there's lots of people listening today that aren't from the UK, but I'm sure that the timelines are similar. So in January last year, the outbreak was first declared, and we received the first sample in the UK on the 19th of January for testing. By February, we'd established a testing capacity of 8,000 samples per week, and that was within the Public Health Laboratory Service, also known as Public Health England. And then from March, we had to stop contact tracing and divert all of our testing efforts into NHS patients rather than community testing, as we had been in contact tracing. And at this point, the NHS put their plans together to provide 25,000 tests a week in capacity. By April, we introduced our Lighthouse Laboratories. So our Lighthouse Laboratories are mass testing laboratories designed just to provide coronavirus testing services. And they started with a capacity of 10,000 tests per day. In May 2020, Test and Trace was launched as a governing body for the COVID response in the UK. And then in June, we rolled out antibody testing. And that was largely used for NHS staff, healthcare worker staff, to look at antibody levels, but it was also provided to the public as well. In June, in July, the number of cases in the UK began to decline, and we thought we had a rest, and we were planning what we were going to be doing for winter. We continued to build our sites, predicting that there would be another peak of infections in winter. And by August, we had 370 different testing sites. In September, for us, largely related to opening up of lockdown conditions and children returning into school, we had beginning of another peak of infections, and not just coronavirus, but there was general respiratory virus infections that are often seen around the start of a new school year as children begin to mix in different groups. The Lighthouse capacity was exceeded, and NHS and PHE offered support into the test and trace system from our mass testing laboratories. And track and trace was implemented. By October, we had 800,000 a day capacity met via the test and trace, so the mass testing laboratories, and that was in addition to our NHS laboratories. And then in December, we reached 1 million tests per day capacity. In December was the first classification of a VOC in the UK. So for us, that was B1117, also known as UK Kent strain. And we began a VOC assurance programme. So in January 21, we began a variance of concern assurance programme to ensure that the tests that we've got in use are able to detect variants of concern. In February, we introduced in the UK international travel quarantines and new testing regulations, so that there was a testing regime in place for days 2, 5 and 8 following return from travel. And then in March, we began a programme of making lateral flow testing available to certain groups, so for frontline workers, people that couldn't work from home. And we also established private testing standards. So for suppliers wishing to provide tests in the coronavirus private testing market, there were some regulatory standards that we've asked them to adhere to. In April 2021, we established sequencing capacity for every single positive sample that we diagnose in our mass testing, our borders and our NHS laboratories. And then just this month, we've launched sequencing and genotype reflex testing for variants of concern, again, for all positive samples in NHS laboratories and Lighthouse Laboratories. So we have a programme where every positive sample with a sufficient viral load will be tested for genotyping and for sequencing at the same time, and those results are matched. And that's a part of a 3-month real-world evaluation to establish where the role of genotyping fits in with sequencing. And I should add that the capacity that we have at the moment is supported by the fact that we have a fairly low prevalence rate at the moment. Should the prevalence rates go up, we may need to review where we're sending our samples and which ones are getting genotyping and sequencing. So what diagnostic techniques do we have? So we began with RT-qPCR, an in-house method, and same in many countries. And I think the common primer probe sets are, for example, from CDC US. And then we moved on to commercial qPCR methods. And then we started looking at other technologies such as LAMP, so loop-mediated amplification. And then driven by consumables and capacity and turnaround time demands, we started looking at other techniques that were faster, such as direct testing, point of care testing. So point of care testing, we would classify as a test that you might be able to perform outside of a laboratory, still by trained personnel. And they're particularly important for our accident and emergency and our admissions, so that we can keep the patient flow going. So rather than waiting 2 to 3 hours for a result, you can have a result generally in less than 1 hour. We've looked at antibody and antigen testing. So at the moment, we're using lateral flow tests widely. And there are also other antigen and antibody fluorescent detection platforms that do require laboratory facilities. We've explored mass spectrometry. We've explored ePCR. We've got antibody testing twice. I'm obviously very keen on that. Antibody testing has not been used largely for probably the last 6 months or so, but now that we're in a stage where we're achieving good coverage of vaccinations, and there's possibility that antibody status could be of benefit to people, then we're expecting that antibody testing will become more commonly used. There's microfluidics, so this sort of test on a chip sort of technology, and then there's also things like organic biomarkers, so breath testing. So at the moment, we don't have any breath tests in use as part of our diagnostic or screening programs, but they are being evaluated. And there's interest in things like organic biomarkers, because you can get an even quick, a faster result than lateral flow testing, and it's quite non-invasive for patients. And then the last one, my favourite, I left the best till last, is scent dogs, so sniffer dogs. So there is some ongoing work in this country and others on the use of sniffer dogs for coronavirus detection, and there's been an evaluation of that as a technology. I'm working in the technology validation group. I'm very excited to work out how we might validate and EQA our sniffer dogs. So the sniffer dogs are really interesting. So they've been trained so far using socks worn by patients with coronavirus. So the socks are taken away and small squares cut out of those socks, and the dogs are trained to use those. And they've had reasonably good sensitivity with positive cases. And specificity slightly less, which means that potentially they could be identifying people that don't have coronavirus. So there's more work to do to understand whether that's useful or not. And there's situations where it's considered it could be useful, so airports, train stations, sporting events. No plans to introduce them as an official testing service at the moment, but definitely under consideration. So how do we validate and verify all of these different methodologies? When we began last year, there was very few positive clinical samples for us to test. And the best way to validate an assay is to put clinical samples through that are reflective of the intended use of the assay. So the instructions in the manufacturer's IFU, instructions for use. We didn't have any quantified standards, so for measuring things like sensitivity of our assays. So we use cultured materials to evaluate the performance of in-house tests in the beginning of the year last year. And of course, this tells us that the assay can detect the virus, but it doesn't give us much indication of how that works clinically. And so as time's gone on, we have established a clinical validation method. A number of in-house assays were approved internationally, but then from April onwards, we moved on to commercial PCR kits and commercial controls became available. And NEQAS began providing us with EQAs to be able to monitor our assays. We found that the commercial kits were equal or superior to in-house methods, and for quality purposes, we prefer to use commercial methods or apply for derogation, so for our regulatory bodies. And to do that, we would provide additional assurance that our assays are working. But the supply issues challenged all areas of our testing. So from every aspect of the laboratory, really, there were supply chain issues, and generally they happened as you resolved one, another one appeared. So we began with difficulties in volumes in extraction kits and not being able to source enough extraction kits for our samples. And then there was a PCR enzyme shortage. At the same time that the UK began to peak, we were a couple of weeks or 3 or 4 weeks behind other European countries, so Italy and France. And by then they were already in full swing of their supply and their testing service. So there was a European-wide demand on products. And some of the primer probes that we sourced for our in-house assays were contaminated or had quality issues, and so the contamination issues were linked back either to production of control material within the same facilities that the primer probes were made, but also they were being produced in countries where there was a high prevalence of coronavirus, and in some cases it was people that were working in those facilities and they had to have complete shutdown and decontamination. And again, we had some quality issues, and that was related to asking for primer probes to be made yesterday and delivered today, and they wouldn't have gone through the usual quality assurance processes that they would do for an established test. We ran out of pipette tips, we ran out of virus transport medium, and in some cases switched to using dry swabs or switched to using saline. We ran out of swabs, or not ran out of swabs, but our validated swabs were in short supply, so we had to rapidly go and validate and source alternative swab suppliers. We ran out of ethanol, or became low on ethanol, and ethanol is widely used in our extraction processes. And then plasticware became an issue. So in particular, the 96 well plates that we use on our high throughput platforms. And then some really simple items like basic plastic pipettes and biobins to put our waste into. So how did we manage to increase our capacity and preserve our consumables? So increase our capacity, a big step for us was CL3 requirement for processing samples. So in the beginning, we were required to process our samples in CL3 and as a minimum inside safety cabinets. So to get around this, people began using heat inactivation. They began using primary lysis buffer tubes. So there's a number of commercially available lysis buffer tubes where the sample's inactivated on addition to the sample, so you can open them in the laboratory without needing additional containment facilities. And reports for those that have been used in the UK or have been validated in the UK in terms of their viral inactivation properties are published on gov.uk. We looked at sample pooling. Sample pooling work paused because the prevalence rate increased, and pooling is generally useful when you have low prevalence. But we did find through those early days that pooling is very operationally challenging in terms of traceability and the size of pools and how much time you save in terms of turnaround times if you have to go back to those pools and retest. So there's still some ongoing work with automated solutions for pooling. We tried direct methods, so bypassing the extraction, to use direct PCR and direct lamp testing. They were generally less sensitive than extractive methods, but still a useful solution in particular situations. We reduced our volumes of extraction, so halving, for example, the amount of ethanol that we used, or reducing the number of wash steps. And this also required some validation. We had, with our suppliers, we wouldn't ordinarily in the UK buy things on national contracts. They would be locally selected from either from our public health laboratory networks or from regional NHS laboratories. So we had suppliers that were saying they had orders coming in from everywhere and they didn't know, which to prioritise. So we set up national groups for supplier relationship managers. And between us and the commercial companies, we looked at what stock was coming into the country and what we were testing at each site. And we became very good at postal services at moving stock around to whichever laboratory needed it, had the greater need, to make sure that we didn't have a break in our supply, in our service. We had daily calls with our national health laboratories and our public health laboratories to ensure that those supplies are moved around. And then behind the scenes, we were working with suppliers to help them in their production lines and source items that were causing them difficulty in production, and also supporting the development of new products. So with such changes going on and inconsistent supply chains, how do we validate and verify and maintain the quality of our tests? So access to clinical samples for some periods for us has been very easy. We've had high incidence of infection, and in this case it's really easy for us to obtain samples either from our clinical laboratories or by a recall system where we would ask positive patients to donate additional samples. We can use biobank samples to validate and verify our assays, particularly for extractive PCR assays. But if we're using contrived samples, for example, for our antigen tests, we need to be really careful that we reflect the samples that the manufacturer is intending for use. So if they're intending that you use a saliva sample, it's really for a lateral flow, for example, it's important that you use a sample that is saliva or as close to saliva as you can get, because there are, for example, for a lateral flow test, the viscosity of a saliva sample is very different to a virus transport medium sample that you keep in the freezer. And that needs to be carefully considered when you're assessing your assays. CE marking for SARS-CoV-2 assays is generally self-certificated, and it doesn't give you assurance that the assay performance is sufficient for your testing service, and it can vary by supplier and the amount of pre-validation work they do before it goes onto the market. And there were hundreds of tests on the market by summer last year. So the method for obtaining the stated performance characteristics that you see, so when we purchase a test and you look in the manufacturer's instructions for use and they state that their test is 99% sensitive and 100% specific, it's really important that labs look at how they obtained that information. And if that was obtained through, for example, contrived materials only at a high viral load, then you should be considering that you need to do more than just a verification before you start using that assay. So typical things that you might find in an IFU for assay evaluation is that the viral load of the samples used to calculate the performance data may not be stated. They may state biosafety for inactivation of samples using their collection buffers. It's really important that there's evidence of that biosafety data. And if there isn't biosafety data, then you shouldn't assume that those products inactivate. And we found through products that we've validated when we've had them independently looked at in terms of inactivation, they don't all inactivate. So it's really important that you look at that. So there's an urgent need to roll out assays, which means detailed validation prior to use can delay the rollout. So there has to be a marked balance between the validation need and the need to put the test into service. So with the technologies validation group, we've worked with some statisticians to work out what the smallest number of samples that we can reliably test to provide a good validation of products. Many NHS and PHE laboratories supported real-world evaluations and validations of those products alongside testing of clinical samples. So when we had products or assays that we wanted to use, we sought help from NHS and PHE laboratories to run those in their laboratories alongside their existing tests and provide a comparison of data. So how can you monitor your performance locally when you introduce these tests? So in the UK, we use a system called UCAS, and aware of colleagues that aren't UK based. So UCAS is the United Kingdom accreditation scheme, and that we use that for our testing laboratories. And it's essentially a standard of processes that a laboratory should go through. And I know that there are equivalents in your countries. So because the COVID supply chain varied so much in the first 12 months, most laboratories were using between 2 and 4 testing options for coronavirus to cover their capacity and meet turnaround times. And often they were used interchangeably. So they may be using a product for 7 days in a row, and then that supply chain became challenged, and then they would switch to another method. And sometimes it was a whole method change, or sometimes it might be the extraction method. So you may use the same PCR assay, but your extraction method differed. So providing assurance and quality control when you're using so many different platforms was a challenge. The ancillary items, including the swab types and sample collection media, varied. And from this, we did some evaluation work with different sample collection media types and their stability over time, and there was variation in those. There was pressure in laboratories to divert work away from our core functions. So we had biochemists working in COVID labs and genetics working in our COVID testing labs, and it further decreased our ability to maintain the routine quality assurances. But then in some laboratories, the drop-off of routine work, for example, routine microbiology samples for a period, became lower. It gave people an opportunity to focus on other areas and quality requirements, including their risk assessments. The natural history of SARS coronavirus was poorly understood at the outset. So there was cases where people would talk about receiving negative PCR results when they had COVID infection. So there was a lack of understanding around the upper respiratory viral load and the lower respiratory viral load, but also that many people would present to A&E after they'd been ill for a particular period, a prolonged period, and by that time their viral load was low or lower, and hence there was some confusion around viral loads and negative PCR results. Some of the tests that we use are qualitative, so we just get a straight positive or negative result, and accurately monitoring their performance or looking at their sensitivity and lot-to-lot variability can be quite difficult. And how do we use these tests? So for coronavirus tests, there are lots of different use cases, and there are different performance characteristics that could be acceptable for those use cases. So extracted PCR remains the gold standard comparator method, but it's subject to frequent debates, and I'm sure many of you have heard those, as to whether PCR is a good comparator for lateral flow tests and whether PCR is the best test to test for infectivity. But PCR is a really established technology, and we've got many years of optimization experience of those assays, and we've got a skilled workforce that are familiar with the characteristics, what to expect and how to identify good results and poor results. And we also have laboratories with equipment to perform PCR tests. So we have the extractors and the amplifiers and the high throughput platforms. It's a common method in virology with experienced manufacturing facilities or suppliers, and a relatively short lead time to make new products. And I'll come to a little bit at, towards the end about variants of concern and producing assays that can detect variants of concern. Because we have good experience with PCR and with molecular techniques, we can rapidly introduce new tests. PCR is extremely sensitive for detecting viral RNA, but it can't tell us whether we've got an acute infection, a recent infection, or a recovered infection. CT values can loosely be used to determine the viral load at the time of the samples taken, and so the CT is cycle threshold, and a low cycle threshold, for example, 5 compared to 25. 5 is classed as a high viral load, and you're more likely to be infectious. But again, it depends on the quality of the sample that's been taken, the type of sample that's been taken. So other methods such as lateral flow testing have been introduced, and these are much less sensitive than extracted PCR. And they are in a camp of people that think they're a really good idea, people that think they're not a good idea, and people that think that they would prefer that their use is restricted to particular use cases. So a good use case for a less sensitive test is in a situation where there's no alternative. So if your choice is a lateral flow test or no test, then a lateral flow test is a good test. If you're in a situation where you're going to be performing repeat testing, for example, asymptomatic staff screening, home testing as has been introduced, the sensitivity of those tests become increased the more frequently you use those tests. As a one-off test, when there's alternative methods such as RT-PCR, then lateral flow might not be the best test to use. And then in general, in the validations and verifications that we've looked at, for both antigen and extracted molecular techniques, that the specificity has been really good. We've had very few assays where the specificity is poor, and they typically range from 95 to 100%, but it's the sensitivity of those methods that are more variable. So what verification standards can we put in? What can we do to ensure the new assays that we introduce in a limited time frame meet the requirements of testing? So we've developed some target product profiles, and in the UK our regulatory body is called the MHRA, Medical Health Regulations Advisory, and we've worked with those groups to develop a set of target product profiles for technologies. So to state, for example, what the limit of detection for an assay should be, what the sensitivity and specificity should be, but also really importantly, how many samples should be included in a validation. So we stated things like the verification should include samples from the dynamic range of the assay. So if an assay detects down to 1,000 copies per mil, then part of that validation should include samples that are around that limit of detection so that we can determine that it does work at that end of the assay. Towards the end of last year, WHO introduced an international standard. So the World Health Organization introduced an international standard for molecular assays. And that can be used to run through assays when you're first validating them, but it can also be used as a routine, regular assurance that your assay is performing in the same way as time goes on, as batches change, or you change parts of your working process, such as your extraction. We should run characterized controls for all of our assays. So typically a testing kit will come with a kit positive and a kit negative. But that kit positive and kit negative is made by the manufacturer, and it might not necessarily challenge the limit of detection of your assay, and it won't reflect a clinical sample. So ideally, you should use an individual IQC, and that should go on each plate. If you're using a plate-based assay, it should be used at least once a shift, or for example, a high throughput platform, or every time you change your reagent. Whole virus controls are the best controls that you can choose for your laboratories, and that's because a whole virus control will work on all the gene targets, so that if you're using more than one method, you'll be able to use the same control for all of your assays. QCs need to be reviewed in a suitable time frame. So when we're running our QCs and we're running them daily, looking at that trend analysis before those results go out is really important, and not to do that retrospectively. So for once a week to have a look at your Westguard rules. By then, 1 week worth of results have already been released. And then each laboratory should establish an IQA, so internal quality assurance. In the UK for us means that we would retest a specific portion of samples, a recommended portion of samples, so anything from 1% to 10%, depending on what the risk is for the assay that you're testing. So by the usual process would be to randomly select a sample that's been tested previously that you don't know the result for. If you can, anonymise it and put that back through the laboratory and compare both sets of results. And then, of course, it would be fitting an EQA meeting to talk about running EQAs. And in the UK, we use a number of different systems, but that does include NEQAS EQAs as part of our implementation and planning and ongoing assurance. And another good finding in the UK for our coronavirus testing is that we establish user groups, and we typically per platform. So, but there was one platform that was used widely across the country. We had user groups where we would invite the supplier, but we would also invite laboratories and share learning, share validation data, share any performance issues that we had. And then when we use our EQAs and we use our QCs and we identify quality issues, it's really important that we know how to manage those. And again, when we're working at pace, it can be really difficult to make those rapid decisions over what we need to do with our quality issues. So the first thing that we need to do is to understand whether it's a systemic issue or a local issue. The reason for that is you may need to inform other users, and the majority of the assays that we use are used across multiple laboratories. And if it's a local issue, such as contamination of your facilities, looking at where else you might be able to send those samples or alternative tests while you resolve the issue that you have. If it is a systemic issue, then you need to escalate, and I'll cover that in the next slide. Quality issues can be really damaging to commercial companies. And I think a lesson learned for us during coronavirus is that sometimes a laboratory or laboratories may think they have an issue that is actually not an issue. But once you've discussed this amongst user groups, it can get back to the commercial suppliers, and there's a need to make sure that you've investigated your issues before you escalate, and to make sure that you keep an open channel of communication between suppliers. And you can use quality reporting networks to share that information. So we have a national quality lead meeting for our National Health Service laboratories, known as Pillar 1, and our Pillar 2 laboratories. Pillar 2 is the name that we use for our Lighthouse or our mass testing laboratories. So this is an example of the flowchart that we advise people use when they have issues with their SARS-CoV-2 assays, whether that's related to variants of concern or general performance. And it's really a guide to steer people in the right direction, so that if an issue occurs that does need to be raised at a more national level, that there's a process in place. I won't read every step of the slide, but I will include it in the slide pack that will be shared with you. Ultimately, any issue that we identify as a performance issue, a significant performance issue, whether it's a batch issue or an assay may have difficulty identifying a variant of concern, we have a yellow card system where people can go online. It's a dedicated coronavirus test yellow card system where you can report assays, and again, that's UK based. So the yellow card system works by, when you put your report in, you'll be assigned a case number, and MHRA will contact that supplier and give them a short time period to respond to the request that you've made, and then from that request they will decide what additional action needs to be taken. Typically it will be supporting the manufacturer through their processes and making sure that any issues communicated. We've had, within coronavirus 18-month testing period, we've had supplier issues typically when there's a new manufacturer and manufacturers that are producing kits at pace. So the normal checks will have been done at pace. And some of those things are related to having to rapidly replace a part of the kit. So a sample tube, for example, that might end up being a leaky sample tube. We found there's a reluctance with laboratories sometimes to report via MHRA. I think maybe they feel it's a really severe way of responding to an issue, but people need to remember that these kits are used nationally and internationally, and when we see an issue, we do need to report them. So if some suppliers have discussed with labs when there's been issues and said that they will report into MHRA, that's also fine. But you should report individually. If you're a lab that finds an issue, you should report individually. If you leave it to the manufacturer, they will likely perform an investigation first. And when they report into the MHRA, they will report the issue from the manufacturer's perspective and might not fully understand the implications for national testing. And if only one person completes a form, so the supplier or one representative of a user, the MHRA don't have any idea of the scale of the issue. And I think the last point that I put on this slide is that you can maintain good relationships with suppliers and report performance issues, and the key to that is communication. So explaining to them why you need to report it, because it may impact other users, and letting them know ahead so that they don't hear from other people that there's an issue with their test, or they don't hear first from MHRA. And then the last couple of slides, I'll just quickly talk through variants of concern. So as I mentioned before, in the UK, we first declared a variant of concern in December last year. There's a lot in the press at the moment about variants of concern. It's a hot topic, but actually evolution of viruses over time is completely expected and it's natural. The reason that we're seeing so many cases of mutations and new variants is because there's still a high incidence of infection, and the more times the virus replicates, the more chance there is of there being mutations. So at the moment, lots of replication equals lots of mutations. Not all mutations are significant in terms of their clinical severity and transmissibility. So a variant of concern might be classed a variant of concern because it spreads more quickly, or because people seem to be having more severe symptoms. And what that doesn't include is that there might be a mutation that is not related to clinical severity or transmission, but may not be detected in an assay or in some of the targets in our assays. So our laboratories are frontline eye to the ground when we see things in assays that might need investigating. Many of our commercial assays use the same primer probe sequences as were designed at the beginning of last year. And many of those assays are based on common primer probe design. So even our commercial kits will have common, similar or almost identical primer probe genes, primer probe sequences in some cases. And that means if an assay is affected by a mutation, then it could affect more than one of our assays. So it's really important that we keep on top of those. In the UK, we've developed a working group to reduce the risk of failing to detect VOX and FUIs in our frontline assays, and in fact in all of our assays. And we encourage laboratories to report anything that they notice in their assays. And in particular, when you have an assay that reports gene targets, 2 gene targets, if there's more than 3 cycles between those gene targets, so you've got an N gene of 20 and you've got an S gene of 30, that would be an unusual presentation, and we would ask for people to report that via their quality systems. The VOC assurance group that we've established is multi-agency, so it's NHS, PHE, MHRA, and members of the devolved administration, Scotland, Wales, Northern Ireland. And we provide assurance at government level, so we report back to ministers on a fortnightly basis for the activities that we've taken, that have been undertaken. We have a portfolio of all of the assays that are used in Pillar 1 and Pillar 2. We've obtained that through our supplier leads, through our quality leads, and through regular updates, regular requests for labs to update their assays. And we have 3 key areas. One is in silico assurance. So again, working with MHRA, we contact all of the suppliers on our supply list, and we ask them to respond to the most recently published variants of concern and to provide in silico assurance. And then we review those responses to make sure that one, there's no risk, so they haven't identified a variant that might affect that a variant affects the assay. And then we have a look to see that their response is sufficient and answers the question, and if not, we'll go back to them. We have enhanced laboratory surveillance, so as mentioned, we've encourage and remind laboratories to report any issues that they see with their assays that they think might be related to variant of concern. And then when there is an issue, or for our antigen tests, we do in vitro wet testing. So the way that we manage this is we have some high containment laboratories that will culture material from new variants as soon as it becomes available and as soon as they can source it. And then they will make that sample safe to be tested on our platforms, and they'll be just distributed to assays that we have concerns around, or for all of our antigen assays. We don't freely give it out to every laboratory, because we absolutely only want to test each platform once and not increase workload for anybody. I've just brought this chart up to show you what a PCR assay might look like if it's affected by a variant of concern. So this is a typical amplification plot, and if you look at the darkest blue line, that represents what you would normally see for a positive result. And if you look on the axis, we've got a CT of, that should say CT of 17. So the earlier CT 17, and then if you look at the orange or greeny coloured line, you've got a CT of 27. And that CT of 27 is something that you might see when you've got a mutation in your target region or your probe region. So the amplification still takes place, but it takes place much later. It's got a much shallower curve. And if you were to get a sample with a low viral load, there's a risk that you might not detect it in that channel. And then the grey line is just to show that we can get other patterns of amplification, which can be normal in an assay, depending on the design of an assay, but it can indicate, when it's not a straight line, it indicates that you might have primer probe mismatches. And we'd also be interested in people reporting those to us. And then I think I've got 1 minute left to talk about EQA for COVID-19. So EQA is a requirement for UCAS accreditation, and we actively encourage all of our laboratories to register their assays as part of their scope for accreditation for their laboratory service. There's an established NEQAS distribution for SARS-CoV PCR, and variants of concern will be included in those distributions. The existing distribution that we have for EQA will also become an end-to-end EQA system for our primary testing, and then for our reflex genotyping assays and our sequencing. And we anticipate that will begin in July. So the samples that are currently going to our primary testing labs, we'll be asking them to put them through the whole process as for a routine positive sample, which is to reflex genotype test the sample and then forward that on for sequencing. And NEQAS are working with GenQA on the data reporting and return of those results. I should say that there are other commercial suppliers of EQA, including QCMD and Instand. I'm not aware that they're doing end-to-end processing, but there are other good commercial suppliers out there. And then we've also helped with the distribution and establishing the needs for antigen EQA. And then to say that if you don't have an EQA for a new system, because there isn't EQA material available, first is to discuss with NEQAS and to discuss with your quality leads the need that you have, and in the interim to exchange materials between your laboratories. So find a partner laboratory and swap samples between you as an interim EQA test. And I think one of the current challenges for EQA is the number of platforms that laboratories have, and establishing how many EQA samples laboratories need. And then just to finish, I was going to ask people to encourage some questions really, and ask people how they think we can reduce the quality risks without compromising the need to rapidly respond and roll out tests before we have things like EQAs in place. And to ask you how you've managed running multiple platforms and different combinations of extraction and amplification techniques. Any improvements that you think we need for EQA schemes, or any gaps that you've identified that we could discuss and think about how we might develop, and feedback really on what you feel has worked really well for coronavirus quality assurance and EQA, and what can be carried forward and remembered should we find ourselves in this situation with a different organism in the future. So thank you very much for listening, and I'll invite Jennifer back to begin the question and answer section. Sorry.
Okay, so thank you so much for that presentation. It was a really interesting talk. We do have a few questions now in the Q&A box, but if there is anything else you do want to ask, please quickly get it in there and we should be able to get to it. I'll now hand over to Melanie Amplet, who is the Technical Services Manager, COVID-19 Lead at PHE for the Q&A session. So I'll hand over to you, Melanie.
Thank you, Jennifer. And thanks, Mel. I think that's been an excellent presentation, and it's been good to reflect actually on what has happened over the last 18 months. I think we're very sort of in the here and now, and obviously there's been lots of challenges for our testing service, and I think you've been pretty integral to sort of setting up a good governance monitoring system for our COVID-19 tests. So if I go over to some of the questions in the chat. So we've got a question from Ayelet Morrow. Thanks for the question. I would like to know what the gold standard is on sample numbers for validation and verification, please. And then an important question, can you explain the major difference in validation and verification, because the terms are sometimes used interchangeably or loosely? Thank you.
Brilliant, thank you. In terms of gold standards, so there is no gold standard for validation and verification, but what we've done in the system that we've developed in the UK, so our target product profiles that are published on gov.uk and are managed by MHRA, is we use a statistician to look at the number of samples that we would need to test to have confidence with a 95% confidence interval that our results were accurate. So for us, we've selected 150 positive samples, 250 negative samples for a validation. If you look on the World Health Organization site, I think there's a similar number. It might be, I think they might have 100 positives and 400 negatives. And the reason there's no gold standard is because it depends on what the prevalence of that infection is when you're testing those samples, so it will move. So for us, we've gone for a middle ground, which will cover low and high prevalence. And then in terms of the difference between validation and verification, so validation is a very detailed look at how an assay works. So it's not just do you get a positive result for a positive and a negative for a negative. It's right down to cross-reactivity, so running samples of different viruses if you're looking at a viral assay to see if there's any interference. It's looking at the linearity of the assay, so performing serial dilutions to see whether you've got good efficiency of the assay. It's I'm trying to think of all the different things that we do. It's essentially a really thorough validation that you might perform if you, for example, if you were using an in-house assay, you would expect to do the full end-to-end validation. In the UK, we would typically only do a validation if we were using a non-CE marked kit. But what we found through coronavirus is that even though kits are CE marked, the performance of the assay is not necessarily reflected in real life compared to what's stated in the manufacturer's IFU. So a verification is what you would usually use for an assay that you've already got confidence in. So somebody else may have already validated it and you've seen the data, or the manufacturer may supply really good detailed information on how they've validated their assay, or you may have used a similar assay before and it's a new version and with a minor change in it. So a verification is usually a smaller number of samples, and it would be a very basic small number of positives, small number of negatives, and then running something in there that you can determine the limit of detection.
Excellently answered. Okay, we've got another question. So how do you think the impending IVD regulations, which mean we will not be able to use in-house assays without intensive validation, might impact on our capacity to diagnose any future outbreaks of a new pathogen?
So my understanding of the new IVD regulations is that there is an exemption in there for exactly this situation. So there's nothing to stop people researching using in-house assays. There's nothing to stop that taking place. But if there was a health emergency, then we can still use in-house assays. There is a stringent and intensive validation process, but for many of us that may or may not be familiar with the early outbreak in coronavirus, there were some assays that were in use that were in-house assays where actually they were later found to be not as sensitive or specific as they could be. And it's a perfect example of why we need to have thorough validation and verification.
Do you have any suggestions where a suitable EQA is not available, but inter-lab comparison is not an option, either due to the nature of the test or the sample?
I'd be interested to know what type of samples there are, because I think there's an answer for everything. So there are options, I think, for EQA. So we've been working with some control suppliers, and I won't name them because all good commercial companies will be respected, but we have been working with the company to develop controls for technologies that we didn't have controls for previously, and we can do that, and if it's a high volume control and there's a commercial interest, then it's highly likely that a company would be interested in taking those up. So if you posted that question and there's a particular sample that you're looking for, if you pop it in the question box, I'll have a look and see whether I've got the answer.
Excellent. How can we introduce environmental monitoring in our IQA?
Oh, okay, so I'm counting the IQA as internal quality assurance single sample to go around, but in environmental monitoring. So environmental monitoring can be introduced at your discretion in terms of how frequent and how you do that. It's a simple case of structuring where you're going to do the environmental monitoring, swabbing that area. So if it's a PCR test, you swab that area. I would probably use a small amount of VTM on a dry swab. You can swab your door. It's really good fun, actually. Swab your door handles, swab your keyboards, swab fridge doors, swab even the sample racks that you hold your samples in, and then just run it through your ordinary PCR. And you would expect that you would see negative samples in most cases, but you will find environmental contamination. And then it's a case of cleaning everything down and starting again. I've in my personal experience of environmental contamination, we made the mistake of finding some amplicons in one room and nowhere else. So we focused on cleaning that room only. But in the meanwhile, people have obviously been going in and out of those rooms. And in the end, we cleaned every room. So I would say if you find contamination, clean everywhere, otherwise you'll start all over again.
Absolutely. And I think in my experience with environmental monitoring, it's a good idea to look at your menu of assays and maybe put a schedule for revisiting those. So over a 6-week period, you want to have maybe all your assays checked for environmental monitoring. And like you say, yeah, once you have some contamination, it does spread. So yeah, a good deep clean of that area is always a good idea. And then to have confidence to re-swab and get negative results once you have cleaned. Yeah, we've all been there, haven't we? Okay, next.
I was just going to say thank you, Mel, for reminding me that there are other viruses other than coronavirus.
Okay, next question. Please, what is the sample stability guidance for samples used for validation from sample collection to reporting? And how long can samples be viable to be reproducible and an acceptable CT value?
I'm going to ask you to break that down for me, Mel.
Yeah, so I'll break this down. So the first part, please, what is the sample stability guideline for samples used for validation? And it's from sample collection to reporting is the first part.
Okay. So it will depend on the method that you're using. So if you're validating a test where it says you must process your samples within 3 hours, then to get a good validation, you need to collect and process your samples within the 3 hours. In terms of stability, if you're comparing two methods, you could use a sample that was 5 days old, so long as you run it through both of those assays at the same time. If you're comparing a test that you got a result for 4 weeks ago, and then you're going to bring that sample back out of the freezer, that would be less accurate. In those cases, I would validate I would use those samples if I didn't have an alternative, but if there was discrepancy in those results, then I would put both samples back through both methods again.
Yeah, and I think that's what we find. I mean, we do validate using sort of material that we know exists in our freezer, but it's to repeat that screening method that was used again, as well as the new methods. So you're not co-founding for anything that might have happened in the time that the test was originally performed and when the validation is being performed. And the second part was, how long can samples be viable to be reproducible and at an acceptable CT value?
When you're validating, you want to look at samples across a range of CT values. So if you used an older sample that had been in your fridge for 5 days, 10 days, and you are running it through both platforms, it's absolutely fine to do that. For stability studies, that's another thing that's included in a validation. So it will depend on which type of virus transport medium you're using, whether you're keeping the sample in the fridge, whether you're keeping the sample in the freezer. So as part of the validation, it's always a really good idea to experiment and test a sample each day after it's been left out at room temperature, after it's been in the fridge, after it's been in the freezer. But typically, I wouldn't, 48 hours, ideally you test everything within 48 hours if they were being fridged. I wouldn't keep most things at room temperature, although I do understand that saliva samples for LAMP seem to be more stable at room temperature than in fridges. So I would check by the method. And if in doubt and you're not going to go back to that sample for a while, it's always better to freeze, but do not repeat freeze for. So we've rolled out sequencing in the UK for all of our positive samples, and the integrity of our samples for good sequencing results is reliant on labs looking after the samples in the primary lab. So if you put it in the freezer and take it out to do a genotyping test, then put it back in the freezer and then take it out of the freezer to separate it off, the more you freeze for a sample, the more you fragment the RNA, and then you're more likely to not get a good sequencing result back. So avoid extremes of temperatures, fridge for a couple of days at the most, and freeze if you want to keep it for a long time.
Excellent. So we've got a question from Dr. Hopkins. Are we supposed to be receiving inactivated samples in the laboratory? So I'm not sure if that's directed to testing in the UK or tested in general. So it would depend on the laboratory. So in the UK, in our NHS laboratories, we can process inactivated or live samples. If you want to use an inactivation buffer, you can. Inactivation buffers would need reviewing in terms of how stable the RNA is. If you want to do on sequencing on those samples, you might need to extract them as soon as they come into the laboratory. If you don't have containment facilities, then using an activation sample, a sample in an activation buffer is absolutely fine. There's some benefits of using live samples. So if you wanted to culture that sample, so you've got live material to for example, a variant of concern that you wanted to share, or you wanted vaccine studies, then the live sample has advantages. But equally, certainly in the early days, the queue of people trying to get in the safety cabinets in our containment rooms was long. We remember it well.
Okay, sorry, so that's a really good Q&A round. Thank you both. But I'm afraid that's all we have time for today. So many thanks to our speaker, Melanie Smith, Melanie Amphlett for the Q&A round, and to everyone at UK NEQAS who's been involved in the organisation and delivery of these webinars. And thank you for all of you for taking time to join us today. I hope you've enjoyed today's webinar. This is the second in a series of 6 webinars that we're putting on over the next couple of months. For more details on the remaining webinars, you can visit our website and register. So UK NEQAS are active on both Twitter and LinkedIn. So if you do social media, please seek us out so you can keep up to date with what we're doing. And finally, once we've closed this session, please do take a few moments to respond to our short feedback survey. So thanks once again for joining us today, and we will hopefully see you next time.